In this video, we're going to talk about the history of life. This is a painting of life as it may have existed about 2.5 billion years ago. And you can see something important is occurring here. These green mats are photosynthetic bacteria, and they are starting to pump out oxygen, and that will forever change the history of life on Earth. And we'll talk about that, and we'll also talk about fossils and some other things. But first, this is Louis Pasteur, and you know him as the father of pasteurization, your milk and your products that get heated up so that they can last on the shelf for a long period of time. But in terms of evolution, he was important because he put to rest this idea of spontaneous generation, the idea of when something died, a pile of dung or garbage or trash, you'd see maggots, insects, worms emerging from it, that there was some life force that left the dead thing and went into this new life forms. And basically what he showed is that life has to come from other life, that you have to have some spores or some tiny little microbes that get in there to contaminate the broth, and that's not just spontaneously life arising, but that it's coming from other life. And this brings up a good point for the history of life. If life comes from life, how did we get life from non-life? So we'll talk a little bit about how that may have occurred as well. First, if we go back to the early Earth, 4.5 billion years ago, it was a pretty hostile place. It was very hot. There was a lot of lightning. There was a lot of atmospheric instability. There was no oxygen, so there was no ozone layer. So UV rays were bombarding the Earth. There was water. There was a lot of water. But because there was no oxygen, the atmosphere was what we call a highly reducing environment. In other words, think about reduction in terms of things bonding to hydrogen. So carbon would be in the form of CH4, methane. Nitrogen would be bound to hydrogen in the form of ammonia or ammonium, NH3, NH4. Sulfur, hydrogen sulfide, H2S. Oxygen didn't appear until the first photosynthetic organisms about 2.7 billion years ago, and that changed things, as we'll talk about. So in this early reducing environment, how did we get, first off, chemical evolution, or the evolution of biomolecules, of these macromolecules, carbohydrates, proteins, lipids, nucleic acids? Well, two scientists, Miller and Urey, set up an experiment, and they basically replicated the conditions on early Earth. So to replicate lightning and this electrical source, they used electrodes, and they ran an electrical current through them to represent sort of a hot water cycle. They had boiling water that then it was condensed and to form rain, and they basically put in the molecules that were known to have been in the early Earth: hydrogen, ammonium, methane, and they basically let this thing run for several weeks. And what they found was, after this time, that these molecules in this reduced atmosphere produced other important biomolecules. Things like amino acids were formed, lipids, simple lipids were formed, simple carbohydrates, and so forth. So they didn't produce life, but they produced what we call chemical evolution without oxygen there to kind of break apart big molecules. That these molecules got reduced and formed bigger, more complex molecules, and then at some point there they would form early protocells with membranes and enzymatic activity. But how do we know about life from the past? Well, we know based on fossils. So these are all examples of fossils: dinosaurs, an early human skull, petrified wood, amber, a leaf print, or a nautilus shell. So a fossil is just any evidence of life in the past. And when you find a fossil, you first want to figure out what it is, and then you want to figure out when did it live? At what point in the history of life did this fossil live? And so if we look at sedimentary rock, this is the Grand Canyon, we can see that sediments here were laid down over millions and millions of years. And sedimentary rock is important because that's where we're going to find fossils. We won't find them in igneous rock, we won't find them in metamorphic rock. It has to be laid down in sediments. So here's an example of sediments being laid down, and we know that the fossils down here would be older than the fossils up here, right? That makes sense as sediment are laid down. We also can do what's called relative dating. If we find a fossil here, right, we know it's probably, somewhere between that fossil and that fossil. So we can look at fossils in relationship to other fossils and determine their relative age. But how else can we date fossils? Well, we can use what's called radiometric dating. We can use isotopes. Since we're a carbon-based life form, an important isotope produced in living tissue is carbon-14. We also have potassium in our bodies, and we produce small amounts of another isotope called potassium-40. Anytime you eat a banana, you're putting potassium-40, this isotope, into your body. Okay, and there are two important ones. Carbon-14 is important, but it's used more for more recent fossils. It has a half-life of about 5,700 years. Now, what is a half-life? Well, a half-life is basically the amount of time it takes for half of that carbon-14 to be converted into nitrogen-12. So in 5,700 years, you will have half the amount of carbon-14. Okay. Now, notice this is not a linear decay. Otherwise, we'd completely run out of carbon-14 fairly quickly. But then in another half-life, we're down to a quarter of the amount. So in about 12,000 years, we'd have about a quarter of the amount of carbon-14. And then in a third half-life, we'd have about an eighth the amount of carbon-14. So based on analysis of carbon-14 in fossil tissue, we can determine about how old they are. Of course, if it's 10, 100, 500 million years old, that's too many half-lives. Carbon-14 won't exist in those fossils. But we do have another one. We can use potassium-40. Potassium-40 has a half-life of about 1.3 billion years. So if we find some fossilized bacterial mats and we think they're about 3 billion years old, we can analyze the potassium-40 in those fossils to see if we're in that area. So let's look at some ways that we would use radiometric dating to age fossils or to date fossils. So here's a sample question. I'm going to let you solve this in a minute. You discover 3 fossil organisms and want to determine their dates. You measure the percentage of carbon-14 remaining in the fossils. Place them in order from oldest to youngest. Okay, so go ahead and look at these fossils. The first one has got about the percent of carbon-14 in living tissue. It's got 38.7%. The second one, 77%, and the third one, about 52.1%. So go ahead and pause right now and see if you can figure that out. So go ahead and pause. Boop. Okay, so fossil A, notice, is well over one half-life, right? If it had 50%, that would be one half-life, about 5730. So this is probably, again, 9,000 years old or so, because it's getting towards a second half-life. Fossil B has 77% of the carbon-14, so it's not even at 1/2 of a half-life. So maybe that's about 2,200 years old. And then fossil C is just about at one half-life, almost one half-life, so it's almost at about 5,700 years. So your correct answer there would be the oldest A, second oldest C, third would be B. Okay, let's try another problem. You discover a series of fossils and want to determine their dates. You decide to use carbon-14 dating based on the table about how old is fossil A. So living tissue has the ratio of carbon-14 to carbon-12 about 1 to 1 million. Fossil A has about 0.74 to 1 million. So go ahead, look at that and hit pause and see if you can figure it out. Okay, so fossil A is about 1/2 of one half-life, right? So it's close to 0.75, so it's about half the way to 0.5. That means it's about half the way to 5730. So that's going to be 2865. Okay. Well, how about fossil B? Let's see if we can figure that one out. How old is fossil B? We'll go ahead and hit pause and see if you can figure out which of those is correct. Okay, so notice fossil B is about 0.25. It's close to 1/4, which would be 2 half-lives. So that would be 1/2 and then another half. So 5730 and 5730 is about 11,300. So this is how we can use radiometric dating to age fossils, and you're definitely going to have problems similar to this on your evolution test, and then probably on the test, the AP exam. So in the next video, we'll go over the sequence of how we believe life evolved over the past 3.5 billion years.